Yeah, thank you for inviting me and for taking up this idea which we created in Geneva last year. All right, the paper I selected is entitled The Effect of Investigator Compliance or Observer Bias on Calculated Efficacy in a Pertussis Vaccine Trial. And I did not select this publication because I happen to be a co-author, but because the findings impressed me very, very much. And the first author and brain behind this study and these analysis is Jim Cherry, an honorary member of our society, a grand figure in pediatric infectious diseases, and I'm sure most of you know him. Now, this is my busiest slide. It's the context of the whole study, and you should know that in 1990, several vaccine manufacturers had developed so-called acellular pertussis vaccines, which contained only a few specific antigens of the organism at a time when the conventional Bordetella whole cell vaccines were thought to cause brain damage in some children, which, by the way, gladly turned out to be a myth on further investigations, and there was pure coincidence and not cause and effect on that. But still, this new generation of vaccines were developed, and they easily were shown to be much better tolerated in terms of local and systemic short-term side effects. But the big question was, how effective would they be? And I, as a young physician, had the privilege to be asked to coordinate one of those trials, namely the one out of Erlangen in Germany. And Jim Cherry and Clemens Stehr from Erlangen were the PIs, and I was the study coordinator. So what we did is we identified 227 physician practices in various parts of Germany, our study sites. Most of them were pediatricians. And the children which we enrolled were healthy 2-month-old infants who received either the Takeda Lederle acellular combination vaccine with tetanus and diphtheria called DTaP, or the conventional gold standard whole cell pertussis vaccine. Or, and this was our control group, DT only without pertussis. And this was ethically sound at that time because in 1990 in Germany, like in many other European countries, there was no universal recommendation for pertussis immunization due to the fact that I just mentioned, the unsubstantiated fear of brain damage in some children. So Therefore, we randomized the infants one-to-one to either DTaP or DTP in at that time, usual 3+1 schedule, or to the DT vaccine in the 2+1 schedule in an open arm. And the assignment to the one or the other stratum was based on parental preference, because some of the parents wanted their children to be immunized against pertussis and others didn't. Now, the decisive point of that study was to detect pertussis cases in order to prove effectiveness of the vaccines. And therefore, we had an active surveillance for cough illnesses with phone calls to families every 2 weeks. In total, there were more than 10,000 infants recruited, and all these 227 study sites had to phone up their families every 2 weeks. And if a cough illness of at least 7 days duration without improvement was reported on the phone in any household member, they were invited to a visit at the study site for a nasopharyngeal specimen for Bordetella pertussis, still culture at that time and not PCR early on, and a serum specimen with pertussis antigen serology as a baseline serum was to be obtained. And then a week later, they received another phone call, and if the cough illness in the index case still persisted for more than 14 days, then the patient was visited by one of 3 central investigators, which I was one of them, from the Central Study Center for a standardized evaluation, and we also took a follow-up serum specimen. Now, we had a very early observation, namely that the cough reports which we received from the study sites were quite variable in proportion to the recruited infants. Some study sites, after a couple months of recruitment, had not reported a single cough illness. Others had reported a few, and some had reported many. So it was Jim's idea then to analyze at the end vaccine effectiveness by the physician compliance with the defined study procedures. And to do so, we separated the physician practices into 3 groups. One was the high compliance group. Those were all physicians with a cough reporting rate of at least 20 per 100 person years, which would mean 1 out of 5 persons had at least 1 cough during a year, or which is approximately the background, was the background coughing at that time. Or referral rate to the central investigator, remember, twice as long cough illnesses, of at least 10 per 100 person. And the low compliance group were those with a reporting rate of less than 10 cough cases or a referral rate of less than 5 per 100 person years. And any study site which didn't fulfill the criteria for high or low compliance ended up in the intermediate compliance group. The case definition for defining pertussis was a positive B pertussis culture or a significant IgA or IgG rise to pertussis toxin, the only specific antigen of Bordetella pertussis, and either at least 7 days of cough, which is considered or was considered a mild pertussis, or the WHO case definition at that time. The typical pertussis means at least 3 weeks of cough with any one of the 3 typical symptoms, paroxysms, whoop, or vomiting. And this is the table with the results after stratification by high, intermediate, and low performance of the study sites. And as you can see by looking at the vaccine effectiveness of DTaP compared to DTP for the mild and typical pertussis, and on the left-hand side, and for typical pertussis, first of all, DTP, the conventional wholesale vaccine, did better than the acellular pertussis vaccine, something which was observed in some of the other trials as well. But what you can also see is that the acellular pertussis vaccine was only 40% effective in preventing mild disease against pertussis. However, when you look at the intermediate and low-performing groups, the vaccines look much better. So the effectiveness now in these study sites, in the same time of the year, same background of the children, was 75 to 78% for acellular vaccine against mild disease, and 83 to 85, not as much higher as with the acellular vaccine. And for typical pertussis, you also can see a better vaccine effectiveness for the acellular vaccine, but hardly any difference for the wholesale vaccine. Now, why is that? This is because when you look at the attack rates in the study children, we are only looking at the household members here, but at the children, you can see that in the high compliance group, 2 out of 100 children had pertussis if they were immunized with DTaP. In the intermediate group, it was only 1 out of 100, and in the low compliance group, it was only 0.5. So there's no reason to believe that if the physician does what he's supposed to do, that then the vaccine would be less effective or better effective. And there's no reason to believe that the true incidence of pertussis would be different depending on the study physician's performance or compliance. But the true reason is that the high compliance group did discover less typical cases and is much closer to reality. There's also a difference between the high and intermediate group for DTP discovery of pertussis cases compared to the low compliance group, and certainly also for the DT group, but not as pronounced as for the acellular pertussis vaccine. So the conclusion of the authors was, and that's the take-home message, that the type of observer bias that we have noted in our trial is likely to occur in all trials, especially if the endpoint that you're looking at is a disease which can have different grades of severity, and only if you do it really correctly you will also discover the less typical cases, and then you will have the true picture, so to say, of the vaccine effectiveness. Thank you very much for your attention.